questions that is been asked. We will answer to them and then we'll stop in 5 minutes. So you can start then recording. Yeah. Once it is live, please let me know. So good evening, all of you. Welcome to the live session of the NPTEL course. So we are almost coming to the end of this course. We are moving on to the last few weeks. And I'm sure that by this time, students have already started seriously preparing for the final exam. I'm sure you are doing well with the weekly assignments. We don't have much of a live questions that has been coming. But there are questions that has been already flagged in the forum. So we will take it up one by one and then go forward for it. So there is a question that has been asked, like when you are working with a non-blocking cache and when you are trying to prefetch an instruction, if it is an L1 miss from L1 cache, we send it to L2 cache, but in the meantime, we can fetch another instruction from the cache. How can we process the other instruction at the prefetch stage when the previous instruction is still waiting for the data? So this is a classical case of when you have an instruction that you need to bring and that particular instruction encountered an L1 cache miss and whose processing is going on. In the meantime, can you prefetch another one? So as long as the second one is not dependent on the first one, if they are mutually exclusive, and the cache is a non-blocking cache, then we can always go with the bringing up with the next one. That is the answer for this. So there is a second question that is asked that P1, in the case of cache coherence, that P1 reads A and goes to a shared state. However, at the beginning, all the caches are empty. It means the read miss is not a shared miss. Some of the P2 or P3 does not have the location. Rather, it's an exclusive miss. Only P1 read the data and that should go to exclusive state. Yeah. So this is a clarification that I wanted to go. In the case of an MESI protocol, the very first processor that is going to give a read request will go to the exclusive state. And thereafter, the subsequent readers only will come to the shared state. So this is the way in which this is going to be moving forward. So first user may not be in the shared, first user will be always there in the exclusive access mode. Then there are some questions which students asked pertaining to the role of MAR and MDR. So one question is specifically on after address decoding in processor memory interfacing slide, we selected one output of 2 power n. Was the selection of output based on input of address decoder? So they are asking what is the correlation between MAR and MDR. So MAR is the register which carries the address of the next memory location from which a memory access has to take place, be it a read or write. Whereas MDR is the register which will carry the data in case of a write operation, or it will get the data after a read operation. So MAR carries address and MDR carries the data. Now there are questions very specifically on what are the ways in which how can you prepare well for the exam. So as far as the final exam is concerned, this is the tips that I wanted to give. 50% of the exam questions would be more or less connected to the assignment questions. So for the assignments, the detailed solution is also given. So when you work with the assignments and you are aware of the detailed solution, then kindly practice it. Similar kind of questions that you will get. 20 to 30% of questions should be from single recall type. So you go through the slides, you understand the concept. We are trying to see whether you could recall the basic concepts or not. And 15 to 20% would be little open and challenging questions. So getting 60 to 70 marks is very much easy if you follow the assignments regularly and you are able to know the answers. So similar kind of a pattern that you can work. So spend regularly time on all the videos, go through it. There are tutorial sessions or problem solving sessions at the end of every week that will also be helpful. And with this, it would be much more comfortable position for all of you, those who are planning to appear for the exam. Exam is not that hard, so please go and write exam. topper students would be given opportunity to work in our research group as interns in the upcoming summer. Okay. So we will have one more live session by the end of this month, by the end of October. At that time, we look forward for seeing you in large numbers. So since there are no more questions, I think we should wind up this session with that. And thank you all for joining.
Okay. Okay. So, welcome everybody. There was some technical problems. That's why we could not be able to see many of you on the YouTube. So, welcome to the live session of Cell and Molecular Biology. And if you have any questions, you can. And if you have any questions, you can unmute your mic, turn on your camera so that we can have a better interactions. Aditi, do you have any question? Okay, good. So I'm sure you are fully aware of the format, right? So in the cell and molecular biology enzyme exam, which is I think in the month of November, we are going to have 100 questions, and these questions are going to be a uh, mix of the some questions would be from assignments, mostly like 30 to 40% would be from assignments directly. Identical questions more of time, and some questions would be based on the recall basics, like whatever we have discussed in the assignments, maybe some value changes or those kind of questions. And then some questions would be related to the theory part, and totally it is going to be similar format what we just now going through, right? So assignments, we have four, we have a objective type of assignments. So the same way we are going to have the NSEM exam also. And overall time will not be a concern because you will get 3 hours. So you have to, very carefully attempt all these questions. Although there is no there is no negative marking, but even then it is good to think about because before you, make the particular options, right? So if you have the specific questions, I will be happy to answer those questions. Okay, so Hania is asking, which topic I should give priority for the exam. So the questions are, being framed from throughout the course. Okay, so I think it is not advisable that you should focus on some part of the course. You will get more or less like 10 questions, 12 questions from each module actually. So we have like 100, we have 12 modules, right? So, and you're going to have 100 questions. So some modules will have like 8 or 9 questions, but some modules will have 10 to 12 questions. So overall, it is distributed throughout the course. So I will suggest you to, go through with all the topics. Then I think Aditi is asking something. Okay, so she's asking that Okazaki fragment in the lagging strand, if you can explain. So Okazaki fragments are the discontinuous synthesis of the DNA, right? So remember that in the leading strand, once the DNA polymerase shifts, right, it do the continuous synthesis and there's no break. But for the lagging strand, the strand has to be open, right? The template has to remain open. Then only the DNA polymerase will come and settle. And because of that, it is actually going to form the discontinuous synthesis. So it is actually going to synthesize the small fragments. And then these fragments are going to be combined by the DNA ligase. So that's why there will be discontinuous fragments which are going to be formed on the lagging strand rather than the leading strand. So I think that is what, right? So yeah. Is it clear? Do we have any more questions? Any kind of queries related to the exam patterns? Vinay, good evening, Vinay. Do you have any questions? So it seems okay. So Hania is also asking about the pattern. So pattern is where so we are going to have 100 questions. You're going to have the 3-hour exams. In the 100 questions, you will have approximately 40 to 50 questions or 30 to 40 questions from the assignments, what we have just given you, right? So they are almost going to be identical, right? And then another 30, 40 questions would be from the questions which are based on the assignment questions, but they will be different, okay? And then remaining 20, 30 questions would be which are going to be on a theory based. Overall format would be the same as what you are attempting right now, like the objective type of questions. And for the, the with 4 options and one of the options is going to be correct. And there's no negative marking. So even if you don't know the answer, you can actually still be able to attempt those questions. So time will not be, it's not going to be a problem. You have to, very thoroughly analyze each and every content, and you have to analyze the sentences or the content of the questions and then the option. And then only I think it won't be a problem for you to attempt all the questions. So I hope it is clear to you. In any case, we are going to have another live session next month. So that time we can discuss more in details. And probably, if you have any more queries, you can drop your queries into the discussion forum also. And I myself, handle those discussion forums, not the TAs. So I will be able to, help you, and I'll be able to address all those queries. So if you have any queries, you ask me. Otherwise, we'll wind up the session. So Manisha is asking if I want to work on RNAs, how I can initiate on it. So I think you're not elaborating what kind of work you want to do with RNA, but and you also didn't say which type of RNA, whether it is a messenger RNA or tRNA and those kind of things. But I assume it is messenger RNA. So if you want to initiate the work on messenger RNA, right? One of the things which you have to start before you even start working with RNA is that you have to keep an extraordinary level of cleanness in your lab, because the labs are, having the dust or other kinds of small, debris or those kind of things will not get you any RNA. And even if you get the RNA, that RNA prep is going to be degraded and not going to be useful even for the RT-PCR or the real-time PCR. If you are going to use that RNA for making the cDNA and you want to use that for cloning some proteins or other kind of experiments, the first and the most important thing is that you should keep your bench, you should keep the working area very clean. And then you can just, work with all the RNA-free solutions like the buffers and waters and all that. And then hopefully there will be no problem. Right. Nowadays, the protocols and the kits are so much advanced that you will not face any problem of isolating the messenger RNA or converting that messenger RNA into the corresponding cDNA and then cloning it into the vector. So it is very, very straightforward what you want to do. But as I said, no, I don't know what you are going to do with RNA. So. Anisha, you have any other questions? Or everything is I have answered? I think I think you give the full detail what you want to do actually. See, I'm not very clear what you want to do actually from a plant or something. You know what you want to do. Can you explain what you want to do? Do you want to clone the proteins? Do you want to see the expression of some gene? Or do you want to do the Nathan blotting? Or do you want if you elaborate, then only I can be able to tell you how much it will be in terms of money and all. Anisha, you are getting my voice right now? If you explain me what is your purpose of doing the RNA-based work, then only I'll be able to, give you more details. Yeah. Send me the Cindy file. As a single PDF file. Where? I sent you the mail. Okay. Now I got it. Okay. So I think I understood correctly, right? You have generated a mutant plant, right? You have generated a mutant plant, and you want to know whether the mutation is being done or not, right? So if I have to confirm the mutations, right? What we can do is we don't have to if it is, and if you already see the phenotypes in your plant, right? That means it is being mutated, right? So the first thing what you can do is, in case suppose when you were doing the mutations, I'm not sure how you were doing the mutations. If it was the random mutations, or if it is a genome-based mutations, right, where you were modifying the genome and then inserting the genome into the plant, then it is a different approach. Or if you are doing the like exposing the plant from radiation and that's and generating a random mutations. So whichever will be the case, for detecting the mutations, you don't have to do a mRNA analysis. What you can do is you can just isolate the genome and do a PCR with the site-directed primers in case you know the sequence, right? And then you you isolate the gene from the wild-type plant, you isolate the gene from the mutant plant, and then you send these for sequencing. And if you do this, right, probably you don't need a lot of money. Okay, it can be done if you are just doing the sequencing. It is, I think, will be done in a very small amount of money. If you don't know the sequences and other things, then it is a different issue. But if see, first thing is, if you could see the phenotypic changes, then definitely mutation is being done. So whether the specific mutation is being done or not, that you will get the information from the PCR followed by the sequencing. Okay, not the RNA. Because RNA is only going to tell you whether the expression of that particular mutant is working or not, and what is the level of expression or not. Because if you integrated that particular mutant gene into the genome, or if you've done the mutation into the genome, then simple PCR followed by sequencing should be good enough. Is it clear, Manisha? Okay, so see, even if you have done the chemical mutations, so chemical mutations are actually not going to be very specific, right? They will be going to do the mutations, but that will be random. So even in those cases also, if you know the particular gene where you are expecting a change, or depending upon, suppose you are looking at the phenotypic changes, right? And from those phenotypic changes, if that gives you an idea about the particular gene which can be get mutated, then you can do the PCR for that particular gene, that particular cassette, or that particular region of your genome. In case the genome sequence are not known, okay, then what you have to do is you have to then I think the process is pretty lengthy, right? But in those cases, and if you don't know the sites of the mutation as well, right? Then messenger RNA definitely will not be an option. Messenger RNA is not going to be. If you know the site, then you have to extract the genome for sequencing to know what the so that's the only. We have any more questions? So whole genome sequencing of the two samples will be very, very costly. So that is something which I will not prefer, and probably you will not be able to do it. The best is go with the phenotype, predict which is going to be get affected, and then do the sequencing of those genome, those particular genes. And if you are lucky, you could find some mutations, then that should be good enough. Do we have any more questions from the students? So I think we don't have any questions from the people who are present in the team, and we also don't have any question from the students who have joined the YouTube as well. So I think it's time to wind up the session, and because you don't have any more questions, and so I'm very thankful for all of you that you have taken the time to join and discuss your problems. In case you want to discuss many more problems or many more queries, feel free to post your queries into the discussion forum, and I'll be happy to address those questions, because I actually handle the discussion forum myself, right? And not the TAs, right? So probably I will be able to give you the very good solutions for your queries as much as I can. And in the next live sessions, we will discuss more about the format of these questions and format of the NSEM exams and so on. So with this, I think I would like to conclude today's live session. In case you have any more queries, post your queries into the discussion forum, and I will be happy to answer. Okay, yeah. So we can wind up now.